This is Dr. Double B, and this is my lecture on MSI, MMR, microsatellite instability, and mismatch repair testing. So the core defect is in the mismatch repair proteins, and kind of a side effect of this is the actual microsatellite instability part, but we'll get into that. So mismatch repair proteins repair mismatches in DNA, like so. That's pretty biologically accurate, I think. And so the four biggest ones we talk about and test for are MSH2, MSH6, MLH1, and PMS2. And so here's a random picture from the internet. So one of the ways we test for these is just by immunohistochemistry. So like all IHC, it's just an antibody to the protein, then with a dye added to actually detect it. So you want this to be positive. You want the MMR proteins to be present. It's DNA repair, so it's nuclear. So A would be normal or intact, D would be intact, and then B and C have loss. And it looks like different tumors are different levels of differentiation here, kind of a well-differentiated nuclear loss, and then maybe a more poorly differentiated tumor. And when you do these, you often have internal controls, lymphocytes, normal epithelium, so you can see the surrounding normal tissues are still intact here, still positive. And then same here, even a case that's intact, positive, you can still see it staining over here. And this is one of those things where negative and positive can be confusing because it's a little unclear. You're talking it's positive, it's there, or it's positive for MMR loss. So we usually try to say intact or loss or MMR deficient. So I have some examples here. So let's look at some colon cancer first. I think it was first discovered and tested for a long time in colon cancer, expanded into endometrial and others as well, and kind of everything now. So here's one that's intact for everything. So when I do this live, I usually start to ask people, but I'll just answer myself, I guess, and hopefully get them all right. So all of these have intact nuclear staining: MSH2, MSH6, PMS2, MLH1. Some of them will be a little bit weaker. PMS2 tends to be a little bit weaker. You can still see it's positive here. So you'd say this is intact and low probability of Lynch syndrome. All right, and here's another one where you have loss of MLH1 and PMS2. And you can see it's not as good here. You see some weak internal control, but you see kind of a better internal control here, showing you that the staining is working, and it is definitely lost here. And so when you have a loss of MLH1 and PMS2, that tells you that the defect is really in MLH1. So these pair up. So if you have loss of MLH1, you also have loss of PMS2, but not vice versa. So you can have loss of PMS2 by itself. And a similar rule applies to the others. If you have loss of MSH2, you'll also have loss of MSH6, but again, not vice versa. So that's how you can kind of figure out which is the actual defect, just by knowing that rule, basically. So most cases of MLH1 loss are due to promoter methylation and not due to a mutation or Lynch syndrome or anything. So the promoters, the CG islands, can become methylated, preventing transcription of the downstream gene. So this is what happens, and you have loss of it. And they talk about methylation testing in the techniques lecture in a little more detail. So again, if it's promoter methylation positive, it's sporadic and unlikely to be Lynch syndrome. And in colon cancer, 50% of these cases also have a BRAF V600E mutation. All right, so here's another case. These three look intact here, but we have clear loss of MSH6 with, again, a good internal control, stromal cells, lymphocytes. And so the rule for most of the other ones is if you have loss, it's higher probability. It's still not definite. You have to do the germline testing. It's more likely than the MLH1 to actually be, syndromic. And now I'll show some other tumor types. Endometrial, which also has a high percent of MMR protein loss. Here's intact. All four have intact nuclear staining. Here's one where we have, again, a loss of MLH1 and PMS2. So if you remember, the main defect here is the MLH1. And In endometrial and other tumors, this is still most commonly caused by promoter methylation, so that would be the next step. Here's a case where we have loss of MSH2 and MSH6. So if you remember the rules, this is a defect in MSH2, so we could recommend germline testing for that. All right, and I actually found cases that had loss for different ones, although I realized later I could have just lied and said, found one with MLH1 loss and just changed the labels around, but these are all legit. So this one is a case of PMS2 loss. Again, you can recommend PMS2 germline testing and good internal control. Now, just to show a different tumor type, this is gastric. Here we have loss of PMS2 and MLH1, so again, the main defect is MLH1. Here there's a little bit of blush cytoplasmic staining, so you really want to see for it to be intact, you want the nuclear staining. And it kind of helps that we have loss of both of these, MLH1, PMS2, so it makes it a little bit clearer in this case. All right, so let's talk about the algorithm. And I've heard there have been questions about this on important tests. So say you have MLH1, PMS2 loss. You can actually either do BRAF testing or MLH1 promoter methylation testing in colon cancer at least, because 50% of the cases with the promoter methylation will also have that BRAF V600E. So if you detect that, if it's positive, you assume it's sporadic and you can kind of stop there and move on. If it's negative, again, that only catches 50% of the cases, so you still have to go to the promoter methylation testing. So usually in real life I would just go to this first, but you may already have a BRAF, you might be doing some molecular testing on the tumor, and if you see that it's BRAF positive, then you can move on at that point. I don't think there's an official kind of best choice here, but again, I would probably do the promoter methylation in real life. And then if it has that promoter methylation, again, it's likely sporadic. If it doesn't, then it's more likely to be Lynch syndrome, and you can recommend germline testing. And a lot of the rules are not always absolute. So even if you had this, you could still, if they had a high suspicion, younger patient, family history, they could still test for it. So that rule about the BRAF only applies to colon cancer. So other cancers, you'd pretty much just go straight to the promoter methylation testing and then use that to decide. So like all things in medicine pathology, you can have weird cases, heterogeneous cases in this case. So here's a biopsy, endometrial biopsy, where it's not the best picture. It was a microscope image and then tried to upscale it, but you can kind of see what's going on here. We have a lot of tumor on both the left and the right. And on the right, the MLH1 is clearly intact. And then on the left, it's clearly lost in the tumor. You still have a good internal control in between the tumor. So yeah, this was a true case of heterogeneity where some of the tumor had it, some didn't. It did have the promoter methylation, and I don't really know which came first. Probably some of the tumor kind of acquired that promoter methylation and so stopped expressing it. I guess in theory maybe you could unmethylate, but again, don't know what exactly happened, but it is heterogeneous, so it would sign it out and say, it's lost, but only in about 50% of the tumor. So that could affect treatment response. So when you have MMR loss or microsatellite instability, you can treat with immune checkpoint inhibitors. The idea is when you have this MMR protein loss, there's going to be a lot more mutations for the immune system to target. So you want to reactivate the immune system with the immune checkpoint inhibitors. All right, so that's the MMR. So that's kind of the fundamental defect. So the microsatellite instability testing, this is kind of testing a side effect of these things. So there are these microsatellite regions throughout the genome that vary between people. Some of them are more consistent between people, some of them vary and are used for things like identity testing. But people just discovered different uses. So one of the uses is in these MMR deficient tumors. So when polymerases copy these regions, they can slip because it's so self-complementary, and you can get small insertions and deletions. You can create this hairpin loop, which basically gives us an insertion in this example. The MMR proteins are supposed to identify this and fix it, but if they're defective, then they're not going to, and so you're going to have these changes in these repeat numbers between the normal and the tumor. So we can do PCR around these microsatellites. PCR, you probably know and was covered in the techniques lecture. And then after that, we run it through the capillary electrophoresis, which sorts things by size and is used for other things like fragment analysis and Sanger sequencing. So smaller fragments go through faster, larger fragments go through slower, and it has 1 base pair resolution. So you can tell if there are a couple nucleotides added or deleted. So when you look at the result of this, it looks something like this, and it's a little messy because we're doing the PCR on these microsatellites, so we're introducing some slipping of the PCR as well, some stuttering. And in a lot of cases, you only see one set, even though you'd think the person might have 2 different numbers of repeats. A lot of them are monomorphic or pseudomonomorphic, meaning most people only have 1 repeat number. Of course, if you're people choose different repeats for different tests. So if they're doing like identity testing, they want things that vary between people. For the microsatellite instability testing, you want things, it's nice to have things that are more often the same. But to be safe, we always test the normal tissue as well. So you can see where that is. And then if it's unstable, you'll see a shift in these peaks. So here's a left shift, so there was a deletion in the microsatellite when it was copying at some point. This was something I'd pulled, or I forget to credit some of these, but this was an example, I think, of a patient with Lynch syndrome and multiple tumors, where all of them were microsatellite unstable, but they all had slightly different changes in the peaks. But so they're all unstable. You're basically looking for a change from the normal tissue. And then here are some of my old examples. So this is a case that's microsatellite stable. We have the normal on top and the tumor on bottom. And we basically see the same main peak and then some stuttering around it. Here's an unstable case. So we have normal and then tumor again with a left shift. So we don't worry about the details too much, but you'd kind of count it out if you want and say like 1, 2, 3, 4, 5. So maybe there's a 5 base pair deletion in that microsatellite in the tumor. And here's one where they're kind of heterozygous for the repeat number initially, and then both of them have a deletion left shift. And then if you look at the top here, these all kind of have just funny names of the different repeat regions, like BAT26 up here. So that's what that is. So another one, normal and tumor. So in a live lecture I would ask, but I'll kind of just go through myself again. So we see a left shift here, so this is unstable. Here, these look pretty similar, so these are stable. Here, this one's a little bit subtle, but you see we kind of have a change. The main peak is still here, but then we see this additional kind of bigger peak to the right here. So this is possibly a 1 base pair insertion, but then the other one to the right over here is a little bit clearer. It seems like we have maybe a 2 base pair insertion here, but this would be unstable. There's another one, looks like a little deletion of a few bases here, and then maybe an insertion here of a few base pairs, so unstable. These basically look the same, so stable. And then here, shifting over to the left a bit, a little subtle, but a few base pair deletion, unstable. So there's an old system, Bethesda, where there were 2 mononucleotide repeats, 3 dinucleotide repeats. Now, I think 5 mononucleotide repeats is considered better. And the rules are, it's MSI high if you have at least 2 unstable markers, MSI low if you have only 1 unstable. And that's kind of basically considered a negative result, or equivalent to stable, where it's not likely to be associated with Lynch syndrome, and it's not likely to be associated with the true phenotype of microsatellite instability, where they have a lot of mutations and better response to immune checkpoint inhibitors. And MSS is microsatellite stable, so all stable. This can also be done by NGS. I had a little of this in the NGS lecture as well. Here we have a mononucleotide A repeat, or microsatellite, and this is a stable one. So you can see that most of them are just kind of the same. Some of them, we see basically the stuttering effects again that we see in PCR. So we see some that have a deletion, some that have an insertion, T base pair insertion, of A's presumably. But this is stable overall, so it's just kind of a little stuttering around there. So here's a case that's unstable where we have a clear deletion. Again, there's some stuttering, so it's kind of some are 6, some are 7, some are 8. So maybe around a 7 base pair deletion in this microsatellite. And here's another example, another mononucleotide A repeat down here. And again, a clear deletion of somewhere around 13 or 14 base pairs with some stuttering around it. So again, one of the main reasons we test for this is for Lynch syndrome or hereditary non-polyposis colorectal cancer syndrome, or HNPCC, and that's germline mutations in these genes. So again, most cases with microsatellite instability are going to be sporadic, and they're just going to be that acquired hypermethylation of the MLH1 promoter. And in colon cancer, 50% of those have the BRAF V600E. So here are just some little stats and things. This one I did remember to cite. You can just see the breakdown of how many cases are associated with each gene. And I think I have another slide on the EpCAM later. And then which will be detected by sequencing analysis and which have larger alterations that could be missed on sequencing. Here's just some more details. I won't go into too much, but just for reference, you can see different frequencies of different cancer rates. So again, colon, so here's the general population on the left here. It'll be very low, but then you have different risks associated with these different genes. Of course, you can see it's much higher, so colon normally would be 2% by age 74, and here it's 44%, 54% for MLH1, and so on. And so if you look at papers and things, you'll find some interesting other causes. So there can potentially be a germline MLH1 promoter hypermethylation. Again, exceptions to every rule. It's maybe 0.6% of Lynch syndrome, at least in one paper. And then you can also have a germline MSH2 promoter hypermethylation. It's caused by an EpCAM deletion because it's the preceding gene to the MSH2. So it'll cause a read-through, hypermethylation, and so this could give you kind of a false negative if you do the germline testing for just MSH2. So then the next step, say you had MSH2 lost by IHC, germline testing was normal, you might still try the EpCAM as well, or the methylation here. This is a random reference because I don't know if it's true, but if anyone knows Chadwick Boseman, Black Panther, and the Marvel movies, he passed away at 43 from colon cancer, very young, and he was diagnosed a bit before that. So even in his late 30s, had a diagnosis of colon cancer at stage 3, which means it was going to the nodes already. And then his brother announced that he had cancer as well. So again, don't know for sure, but just hypothesize maybe it's Lynch syndrome, or it could always be something else. Alright, I threw in one extra thing. This I don't like to talk too much about specific platforms, but this is one I worked with a little bit, and it kind of had an interesting, different approach. So just to show kind of a slightly different technique and how this could be done. So say we have, again, a microsatellite mononucleotide A repeat, and it's being copied in the cells in the tumor. The sequence can slip, the polymerase can slip, and then we have a change in the repeat number. So we had a 1 base deletion here. So now we had 1, 2, 3, 4, 5, 6, 7, 8 originally. Now we have 7. And then in the animation, I'm just going to copy it one more time so it's back to the A again for when we look at it. And so here's where we get into the actual assay. So that was all in the actual patient cells, the tumor. So we had this deletion, and now we want to detect it. So in this assay, they discovered or looked for a bunch of microsatellites that were monomorphic. So basically, they're the same among people, and ones that are prone to this 1 base pair deletion. So they just want to kind of keep it simple, not have to worry about tumor normal, and have a very kind of clear way to detect it. So here's where it gets interesting. So we have these different probes here. So it's a melt curve analysis, but we'll get to that later. So they have this kind of hairpin sequence here. So it's self-complementary here, which keeps the reporter and the quencher together. So it's kind of like when we talked about real-time PCR, which is the techniques lecture. As long as the reporter and quencher are nearby, the reporter is not able to fully fluoresce. But the middle sequence in this hairpin loop is going to be complementary to this one base pair deletion specifically. So they're probing for that deletion. And when it binds, it separates the reporter from the quencher, so the reporter fully fluoresces and can be detected. So if you remember the melt curve basics, it can still bind usually when there's like one mismatch or so. So the one that's wild type up here, it can still bind, but it's going to be a little bit weaker because it's not as good of a match. So then we're doing the melt curve part. We're slowly heating the mixture. The imperfect match, the wild type, is going to fall off sooner. And then the mutant, the one with the one base pair deletion, is going to fall off later. And so you can look at the different melting curves and figure out which it was, wild type or mutant. And so again, I showed some of these slides already in the techniques lecture. But what you're looking at is the fluorescence to time or temperature. It's going to fall off when the probe is falling off as it's heated. And the temperature time is going to depend on if it's a perfect match. So the perfect match in this case would be that one base pair deletion. And then, of course, when we visualize it, and we can't really visualize it in this platform precisely, they have a lot of algorithms built in that do it. But when we visualize it, we usually look at the change in fluorescence with time, because then you get this nice peak. It's kind of the derivative, if you remember your calculus. And so you see a peak when it's falling off. So again, the green would be the perfect match, in this case the deletion, and then the red would be the mismatch, the wild type. And then, of course, in real life, in tumors, everything's always a mix, so even in a positive case, you're probably going to see something like this. Again, the software doesn't really show it for this case, but you get results back, instability scores for all of the different regions they're looking at. So they have these mononucleotide repeats, microsatellites, in these 7 genes. And then you get a microsatellite or an instability score for each of them, and then if it meets certain thresholds, it'll be called positive or microsatellite unstable. So 1 through 4 were just examples of positive cases, 5 through 7 were negative. So again, since I brought it up, main advantage is it's a lot faster than other assays. Disadvantages, it was developed in colon cancer, and it has a little bit weaker sensitivity in other tumors like endometrial, but when we were validating and studying it, we found that by adjusting some of the thresholds, you can actually catch a lot of those false negatives and make them true positives, or at least make an indeterminate category and improve the results there, but won't go into detail on that. So that's all for this one, and stay tuned for more.